Good morning. I am Jacopo Grisotto. I'm a PhD student in the laboratory of Professor Giovanna Paolone, and I'm here today to present you my work and my PhD project that focuses on the neurological effects of exposure to pharmaceuticals in water environment. Basically, as a matter of fact, pharmaceuticals are not completely digested by the organisms, so either humans or animals, and after they have been ingested, they are able to reach the water environment. Little is known about the effect of this persistent presence of pharmaceuticals in environments. And in this case, we want to create a bridge between the ecopharmacovigilance, which is the analysis and field that focuses on the presence of pharmaceuticals in environment, and neuroscience, so to analyze which is the effect of this presence of pharmaceuticals in environment. In this way, we focus our research using the zebrafish model, which is very useful, also because the high homology of the brain areas between humans and zebrafish, as represented here in the bottom, we see that all the areas in the human brain are represented also in the zebrafish brain. And our main objective is to collect the data of environmental chemists that detect and quantify the presence of pharmaceuticals in environmental matrices like waters, lakes, or seas. And we take this information and we recreate the environmental condition in our laboratory. So we perform behavioral tests and histological tests. So we evaluate the tissue damage that is present in the brain of zebrafish. And we do it after the exposure to environmental conditions. I brought here some examples, like a behavioral test that we perform that focuses on the preference of stressed animals to spend their time in darker environments because they feel they are more safe. And instead more exploratory animals, so maybe animals that are not stressed, that are not in a condition of anxiety, they tend to explore the environment going into the lighter portion of the tank. I brought also an example of histologies, and on the left we see a coloration, and in blue we see the nuclei of neurons, these small blue dots. And all the red dots instead are cells that are dying. So by doing this analysis, we are able to understand the extent of the damage to the brain after the exposure to these compounds. The main results that we obtained until now is to detect that diazepam, which is an anxiolytic drug, so a drug that reduces the anxiety and increases the quality of life, alters the stress levels differently in males and females. In fact, what did we do? We exposed the animals to diazepam, and we saw that after the exposure, females tend to prefer the light environment, so they feel more free to explore and less anxious, less stressed. Instead, the males exhibited the opposite effect. So they tend to stay in the darker environment, to prefer the safe place, and we assume that they are stressed. One important approach that my research wants to carry out is the One Health approach. So all the fields that are depicted in this picture are interconnected. So they are they share some importance. In fact, if we moderate the consume of pharmaceuticals and we moderate the consume of pharmaceuticals also in animals, we lead to a decrease of the presence in environment of pharmaceuticals, obviously. Also, the correct disposal of chemical products leads to a better ecosystem, and better ecosystem and healthier animals lead also to an increase in the quality of life of humans. So the main message that I want to bring you is that we do not have only to consider the health of humans, so our health as important, but also the health of ecosystems and animals that lead to better health also of ourselves and of the whole ecosystem. Thank you. And this is the end of my presentation.